Hello, welcome to another screencast, and we continue our discussion of the immune system here, and we'll be discussing now the humoral responses that involve B cells, plasma cells, antibodies, memory B cells, memory helper T cells, and other T cells as well. And I think we'll split this. We'll do that first and then talk about the second exposures and the basis of the vaccines, and then a little bit about allergies in another screencast. So we're going to, again, just focus describing and explaining this humoral response in these cells over here. So here's our roadmap, and we have this first exposure to the antigen or the pathogen. In a previous screencast we had looked at the cell-mediated response, but I want to go over here and look at this humoral or antibody-mediated immune response. So we have our pathogen on first exposure, and we looked already at this pathway here and this pathway. We want to go over here. So there are these other lymphocytes called B cells that free antigens directly activate here these B cells. So that must mean that these B cells have receptors on their surface for for these antigens or these pathogens, right? So this is why this is not cell-mediated. There's a direct activation here. The pathogens are in the bloodstream, and so the humor, the four humors, one of them was blood, and so this is why this is the humoral response. So these B cells here are directly activated, and when B cells are directly activated, they give rise to, and this again is going to be mitosis as well as differentiation. I think I had mentioned that gives rise to over here, and I had just written mitosis, but also this differentiation. You're going to get more of the cells, but then some of them become plasma cells, and some of them will become memory B cells. That is these B lymphocytes here. These memory B cells are just going to kind of hang out, and it's the plasma cells that we are interested in, because when they give rise to these plasma cells now begin to secrete antibodies. And now what do antibodies do? Well, these antibodies will defend against extracellular pathogens by binding to antigens and making the pathogens easier targets for phagocytes and complement. This is what's going on here. So we have, again, binding of things together that is going to cause a response, right? So here we're going to bind antigens. These antibodies bind the antigens and make it easier for phagocytes and other cellular enzymatic processes here to get rid of it. Okay, so it might be a good idea here to pause now and review that. And so let's talk a little bit more about this humoral response. And so what you have to assume is already, and this is a discussion for another time, is that inside here we have a variety of B cells that exist already. So we're just going to go with this assumption, right? And that these B cells are all different. How are they different? Well, the antigen receptor is going to be different. So I have this one, and then here's a receptor, and here's a receptor, right? And so we already have this stockpile of these varied B cells. Why or how, again, another discussion for later, but let's assume that we have these. And so these B cells here on exposure, on first exposure to these antigen molecules here, first exposure to these antigens, will bind to these receptors. And again, now we have binding, and that's going to cause some sort of response in this cell here. And the response is to undergo mitosis, and then to undergo differentiation. And so what we saw is that we get these memory cells, and again they are clones because of this mitosis here. And so we get lots of these memory cells that are going to just kind of be stockpiled and waiting. But then you also get a clone of these plasma cells, and so these plasma cells now then start to churn out antibodies. So that's an antibody right there. And you notice that this cartoon of this plasma cell looks different than these memory B cells. These memory B cells look like these B cells up here, but these plasma cells don't look like these cells up there because they don't have the receptor on them. the job of these plasma cells, the response here of these cells is to churn out antibodies. And you see all of this stuff here. This is supposed to be endoplasmic reticulum, indicating that there is a lot of gene expression going on here. You're getting transcription and then translation, transcription, translation. Lots of gene expression to get these antibodies to be produced. These antibodies are proteins. So let's talk a little bit about those antibodies here. So an antibody is a protein. It's a Y-shaped protein. And there is a business end and a non-business end. So the thing that the antibody binds to is the antigen. And this is the origin of the word antigen. So an antigen is anything that is going to cause a plasma cell to generate antibodies. Right? So anything that is antibody generating is known as an antigen. So antibody generating is an antigen. So this is where we get the term antigen from. Pathogen, incidentally, so you have a pathogen. A pathogen is anything that generates pathos. Right? Pathos or abnormality, right? Pathology, sickness, illness. I should really know what pathos means. I forget right now because it's late. I'll go look that up later. But it does not mean illness. It means something else and I can't remember. Sorry. Anyway, back to the structure of these antibodies here. So this is supposed to be a protein chain down here, these Y-shaped proteins. So all antibodies have this heavy chain right here and have this light chain, and then you have these disulfide linkages that are going on right here. So this is a tertiary structure, that's a tertiary structure, this is a tertiary structure, right? And then this is what's common to all these, right? You have this heavy chain, this light chain, but then on the ends of them you have this variable region, and this is what the V is for, and this is where the antigen is going to bind. So this is a variety of shape. And so if I vary shape, I get complementarity here, totality, and you have different antibodies that are going to bind to different antigens. So here we say that this shape is different there, this shape is different right here, this shape is different right there, right? But all of them do have this characteristic Y shape to them where they have a heavy chain and a light chain. Okay, so there's a little bit about the structure of antibodies. Now, these antibodies come in like 5 different major classes, right? And these are sometimes, antibodies are sometimes also referred to as immunoglobulins, right? Globulin here being a globular protein, right? Associated with the immune system. So the immunoglobulins is where you get like Ig, so immunoglobulin M, IgG, IgA, IgD, IgE here, and this is what they look like, and here's some information about them and the different characteristics. You are in no way responsible for memorizing all these things, but this is interesting because you might have heard of some of these things. So for example, IgG is the most abundant in the circulating antibodies, readily crosses walls of blood vessels. IgG also crosses placenta, confers passive immunity to the fetus, right? And so if you're talking in anatomy and physiology, we talked about conditions where blood types, positive, negative blood types, this has to do with the antibodies crossing placentas and things like that. Okay, so interesting maybe to pause and read that there. Anyway, how is this working? This is again the detail here of going down the center and then to the left. So let me just fast forward here again. So on first exposure, what's happening is that those B cells again are being activated, giving rise to the plasma cells as memory B cells. We've already done that, and then we saw the secreting of the antibodies. Well, this is also happening when you get the cell-mediated response as well. These helper T cells are going to stimulate these B cells via interleukins as well as cytokines. that were actually happening over here as well. So that's what this slide is showing you. So we activate these helper T cells. These helper T cells then proliferate, and here's interleukin-2 and other cytokines that are going to signal these B cells. And so I get this activated helper T cell right here. This is helping with the immune response. And so again, I get antigen fragment there. Right? Some of these B cells, right, will actually, here they show you the binding of them. Here's the B cell took in the pathogen or the antigen, right? It engulfed it after it bound it, and then it's going to present antigen to these helper T cells. When that happens and you get this docking, this binding, those B cells are then signaled to undergo mitosis and then also to differentiate into these memory cells as well as the plasma cells. Those plasma cells then begin secreting lots of antibodies down there. Right? So let's go see what the antibodies are actually doing. Well, the antibodies are binding directly to, in this case we show you binding directly to the virus or to the bacterium, and then the binding of the antibodies actually blocks the virus or the bacterium from infecting cells. Okay, so preventing the virus or the bacterium from entering the cell is one role of antibodies. Antibodies will also here cross-link several bacteria together and again help to prevent them from infecting other cells. So this agglutination, this clumping of them all together. You can have these soluble antigens that are dissolved in the plasma, dissolved in the fluid, and when the antibodies bind to them, it actually brings them out of solution, and then you get this cross-linking going on, cross-link, where now these individual antigens here cannot enter cells and infect them. Right? And so the binding of the antibody to these soluble ones or to the bacterium or to the virus actually now enhances and signals this phagocytosis by these macrophages, right? And so the antibodies, you can think of them as kind of tags or excuse me, tags or signals for the macrophages to come and bind to them, right? And so here's the heavy chain part binding to these macrophages, and then the macrophages engulf them and chomp them all up, and you no longer are infected with viruses or bacteria, etc. Okay. Another component here is that the antibodies will bind to foreign cells, and so this is the basis sort of tissue rejection. So if you have an organ transplant or cell transplant, the antibodies are binding to antigens on the surface of that foreign cell, and then that is in some way triggering this complement. Right? This complement here is an inactive enzyme that then, when it becomes activated, actually creates this lesion or this pore in the foreign cell, and in that foreign cell, the contents of it are removed, the cell lyses, and then that foreign cell dies, and you end up rejecting that organ or tissue in that manner due to antibodies being present. So again, we've discussed here this humoral response down this side. We have not addressed the memory cells in the second exposure, which I'll do, I think, in a separate screencast here. So let me just stop there, and yeah, see you on the next one.